Good afternoon, everyone. I hope that you guys are having a good start to your weekend. I hope that you're having a good Wednesday. Um, so far, mine's been all right. One of my dogs has had some GI issues. That's gastrointestinal. So I had to get a rug out of the house because anyway, enough about my dog's bowel movements. On to our class. What we are going to be talking about today is we're going to be wrapping up kind of the nuts and bolts, the important parts about protein structure, and then we're going to transition to talking about protein analytical techniques. So on the slide deck that we have here, we are looking at secondary structure prediction. So just as an important recap, we've got primary structure, which is just the sequence of amino acids from N termini to C termini. What amino acid follows what amino acid and everything like that. Another important part about that is when it comes to the primary structure of a protein, you have lots of peptide bonds. Those peptide bonds, regardless of the pH, are not going to have a charge. They're going to be neutral. If anything, there's a partial charge, and so they're going to, and they're also going to be great for hydrogen bonding, but they're not going to adopt a full-on charge. So we could be talking about a peptide at a pH of 1. We're not going to account for that that peptide, that peptide bond whenever it comes to like thinking about the overall charge for a molecule. Now, when it comes to oh, word, sorry, when it comes to protein structure, there we are. And thinking about our different levels of protein structure, we've got the primary structure that's going to be stabilized by exclusively covalent bonds. So I'm going to write up here one, and then it's pretty common to see one naught, that NaUGHT, that 1 degree, to indicate your primary structure. So that is going to be stabilized by peptide bonds. But if you want to take it a step further, a peptide bond is an example of a covalent bond. Now, our secondary structure is going to be backbone hydrogen bond interactions. Now, it's important to remember that the secondary structure is backbone. So I got a private message that asks, did we get the starting questions? They didn't pop up. Wanted to make sure it's not my device. No, we have not had those yet. We'll have those when we get into the new material for the day. Or not the new material for the day, the next block of material. So you haven't missed anything. So secondary structure is backbone hydrogen bonds. It's an important distinction to make because when we start talking about the R group interactions, there are R groups that are capable of hydrogen bonding. Now, just as like a little bit of a mental exercise, it might be helpful to think about your proteinogenic amino acids and think, well, which ones do I think could potentially participate in hydrogen bonding? Do you think that your non-polar amino acids are going to be involved in any sort of hydrogen bonding, or those non-polar R groups? Chances are slim on that, pretty much none. Um, so then that leaves us with our polars. Okay, so one really good candidate or one good set of candidates are your polar neutral or your polar uncharged residues, specifically serine and threonine. Those are good candidates for hydrogen bonding. That's, of course, talking about tertiary structure, talking about those R groups interacting. But we're still talking about secondary structure. Now, our secondary structure, one of the very important things that happened in the early 1970s was two researchers, Chow and Fassman, wrote a review article. Could everyone give me a one in the chat if you're familiar with a review article? And if you're not, then either type nothing or some or anything else. Okay, so review articles in the event that you're anything like me. I remember the first time I had to read a review article, I was like, man, this is difficult. This is entirely challenging because I feel like every single sentence in this review article is like chock full of information. I feel like that's almost a paper unto itself, and it is. So in the event that you're like me and you're like, man, I struggle to review articles, yeah, don't don't carry any guilt or shame or anything about that because they are very dense. Now, with that said, Chow and Fastman, they wrote or they made a prediction algorithm, which continues to be refined and used to this day. Now, essentially, what they did was they looked at all of the papers that were currently available on protein structure. And keep in mind, this is in the early 1970s. I think it was like 1972. Yeah, I'm pretty sure it was 72. What they did was they looked at all of these articles that had published results of protein structures, and they just kind of looked through them and they were like, let's see. Whenever I look at this protein structure, I see this region right here. That region has, and I did not draw it as well, but that has an alpha helix. And when they looked at that structure and they looked at the amino acids in that structure, they said, okay, well, here, I'm going to write the sequence of the amino acids that are in that alpha helix. So they wrote them all out. They'd say like A, R, um, M, K, just kind of wrote the sequence of them out. And essentially what they were doing was they're collecting data there. They were saying, okay, well, these amino acids are found in this alpha helix. And they did this for a huge number of papers where a protein structure was published. And they did that for the alpha helices and the beta sheets, and they ultimately came up with basically a scoring system. And that is shown in this table right here. They came up with a scoring system and gave a scoring range from 0 to 2. Every amino acid was given a score of 0 to 2. And this is essentially what's known as a propensity score, where you can kind of think about it as likelihood. What is the likelihood that any of these amino acids is going to be found in an alpha helix or a beta sheet? Now, look at this column right here. And could anyone tell me with a single letter code an amino acid or several amino acids that they think are likely to be found in an alpha helix? So essentially what they were looking at and what they did with this, Michael, arginine. Absolutely. And I know that some of y'all were probably looking at this and thinking, okay, we've got a 0 to 2. Is this 0 is good, meaning it's likely, or is 2 good, meaning it's likely? And in this case, the higher the number, the more likely that amino acid is to be found in an alpha helix. So Michael said R for arginine. Yeah, with a score of greater than 1, it has a greater likelihood of being found in an alpha helix, especially if you compare arginine alpha helix score and its beta strand score. I got a private message of A for alanine. Yeah, absolutely. Someone's saying they can't see my screen. Is anyone having trouble seeing the screen that I'm sharing? Okay. Well, the person who just sent that to me, I think you might need to. It seems like it's on your end. I'm sorry to. Okay. I'll enable that. Okay, so essentially what this is a mechanism or it's a tool that you can use to basically assign a score to every amino acid. And based on that score, you can kind of construct an alpha helix or a beta sheet. The more amino acids that you have that are a 1 or greater, and the higher they are and closer they are to 2, that's an indication that those are likely to be found in an alpha helix. There's also a beta strand or a reverse turn. Now, I want to go ahead and mention this one, this reverse turn, because this is also a secondary structure. It's maybe not as commonly discussed and maybe not as common or as significant as an alpha helix or a beta strand. But this is just a matter of whenever you have a beta strand, if you remember, I drew a structure that looked like this. you have these kind of turns and these movements in the structure that, well, that those can be these reverse turns. So I like to focus primarily on the alpha helix and the beta sheets, though, and really emphasize that the higher these scores are, the more likely these amino acids are to be found in an alpha helix or a beta sheet. Now, I'm not expecting you to memorize or know these particular scores, but I do want you to be able to look at this and say, oh, okay, well, that's what that means. Now, I'm not going to be assessing whether or not you've memorized this or know this specifically. Instead, what I'm going to be looking for is I want you to understand some sort of general rules. One thing that we talked about previously was that the R groups, the amino acid side chains, they're not going to drive the formation of a secondary structure, but they can absolutely disrupt it. Now, when it comes to an alpha helix, I just got a private message. You have not missed the starter questions. And anytime you show up late, even if you miss the questions, remember I mentioned that if you get the starters or the closers, and then the mids, I'm looking for you to participate. If you miss the starters, then hang on. Make sure that you do the closers. So, okay. And yeah. So the starter questions will be up in just a little bit. Okay, so when it comes to an alpha helix, the more likelihood or the propensity or the preference, I guess you could say, is to smaller and less bulky amino acids. So when it comes to like analyzing an alpha helix, you're more than likely going to find something like an alanine, a glutamic acid, a methionine, a leucine, or a phenylalanine. Now, you're less likely to find something like a proline, a histidine, arginine, or a glycine. Now, I've got proline and glycine in bold, and that's for a reason. But before we get to that, when it comes to the beta sheet, there's a preference for larger, bulkier residues. Wait, can you repeat what you mentioned earlier? R groups won't drive the formation. Okay, thank you very much. I'm happy that you asked that, Natalia. The side chains or the R groups will not drive the formation of a secondary structure, but they can absolutely disrupt it. Okay. So when it comes to the beta sheets, you got larger or bulkier side chains. So a tyrosine, a tryptophan, a phenylalanine, methionine, isoleucine, valine, threonine, and cysteine. And I think that it's fair to note that phenylalanine kind of has, well, it's kind of found in both alpha helices and beta sheets. And if I remember correctly, the propensity scores, yeah, for phenylalanine, it's 1.16 to be found in an alpha helix and a 1.33 to be found in a beta strand. So it's possible to be found in both. Um, just so Natalia, did I answer your question? Okay, great. So then what amino acids are less likely or least likely to be found in a beta sheet? Proline and glycine. Huh. So phenylalanine, it's kind of capable of being in an alpha helix or a beta sheet. Phenylalanine just kind of likes to be in a secondary structure. Okay, got it. Proline and glycine, those two amino acids are not likely to be found in an alpha helix or a beta sheet. Now, whenever I think of proline, I always think, ooh, one of the amino acids I struggled to draw as an undergraduate. And now I think about, okay, I like to draw my backbone for every single amino acid. I'm not going to draw all of the atoms, but I'm going to draw kind of my general backbone, NCC. So NCC, then I'm going to go ahead and make the R group for proline. The R group for proline makes a five-membered ring. Oh, why did I write? I wrote the number five because I was saying the word five while I was writing. Big mistake, Joel. Two things at one time. Okay, there's my five-membered ring for proline. Now, proline is the one amino acid where the R group kind of circles back to the N termini. It makes this ring. What that leads to is a very rigid structure. So this R group or this amino acid doesn't like to move very much. Now, if you remember last time, we were talking about a particular bond that doesn't like to move very much. Does anyone remember the bond that doesn't do a lot of rotation? If you could add it to the chat. I'm seeing, yeah, not a pi bond, starts with P. I'll give you a hint. It's between two amino acids. The peptide bond. Absolutely. Great job. The peptide bond is really restrictive. It does not like to move because it's got about 60% double bond character. So our peptide bond doesn't like to move. If you look at this R group for proline, and please do not hesitate to say, wait a minute, that doesn't make any sense to me. What I'm going to do is imagine, okay, we've got the N termini of this proline. Upstream of that, or to the left of this nitrogen, that black bond that I drew right there, that is my peptide bond. Now I'm going to draw the C termini of the preceding amino acid. Okay, could you give me a one if you can, visualize what I'm drawing, what I'm continuing to draw here, or a zero if I don't follow. I know I picked two keys that are right next to each other, but I'm seeing lots of ones, so I'm feeling good about that. Okay, so when you think about a proline, I'm going to put an X over a very restrictive bond. Booyah! Very restrictive. Now, this N to C is typically going to be a phi bond or a phi. It's a phi bond. I've always heard it phi when I'm in a physics or a chemistry class, phi whenever I hear it's a Greek organization on college campuses. But there's my phi bond. Now, that typically has some rotation to it. But in the case of proline, it's much more rigid and restrictive. So proline has this limitation. And so whenever you approach proline and you speak to proline and say, hey, you know what, we're going to put you in an alpha helix. Proline says, no, not interested. It's much like my father-in-law wanting to do something new. No, not interested. Hey, look at this food that I made. No, not interested. Okay, so proline has that rigidity and that restriction. Now, glycine. Okay, so proline is super rigid. Glycine, by comparison, if you think about glycine, I always remember this from when I was a graduate student. I'd give quizzes. And I remember this as just a big, goofy mistake by me. One of the quiz questions was draw an amino acid, any amino acid, any proteinogenic amino acid. And you know what I got more than anything else? I got this right here. Okay, so there's my backbone. And then everyone would be like, boom. Yeah, there we go. Perfect. Hydrogen. It's my favorite R group. That's your glycine. They'd always draw glycine and they'd label it alanine. But at any rate, the point is that glycine, by not having an R group that's got any mustard or anything to it, it's entirely too flexible. So proline and glycine, neither of them like to be in secondary structures, but for opposite reasons. Proline is too rigid, glycine is too flexible. And I'm writing it in this box, but it applies to the beta sheet as well. So I want you to remember, and if you're thinking like, okay, well, what's he going to assess us on about amino acids and preferences or likelihood to be found in a particular structure? I would lean on, well, proline and glycine are my two outliers. They don't like to be, they consistently say, no, thank you to a secondary structure. Then I would follow the general rule or this sort of general idea that smaller and less bulky residues prefer alpha helices, where your beta sheets prefer larger, bulkier side chains. Okay. Now, okay, so that's kind of the takeaway there, too rigid, too flexible. Now, when it comes to prediction, so this is a huge thing because now all of a sudden you can take a sequence and put it into a simple computer program. If you Google secondary structure prediction, you'll find a tool that if you plug in a peptide sequence, it will give you an idea of, hey, this region is likely to be an alpha helix. This region is likely to be involved in a beta sheet. Um, so when it comes to predicting secondary structures, there's some things that you can remember. Glycine is too flexible, proline is too rigid. Now there is hydrogen bonding that takes place in the backbone, and there are other factors that do contribute to whether or not a secondary structure is going to form, particularly what's the solvent, what are the environmental conditions, and then how long is the peptide. If something is short, meaning it's less than six residues, it doesn't really lack, or sorry, it doesn't have the stability to form a secondary structure. And keep in mind, does anyone remember approximately how many amino acids are in the average? This is an average alpha helix. And this is 3.4. Okay. So this is the number of residues in an alpha helix is about 10. Now, one of the things that gets tricky about that is you remember, okay, there's 10 AA in the average alpha helix. Okay. How many rotations is that? Well, it's about 3.6 amino acids per rotation. And so then you're looking at about two and a half rotations. That's just kind of going back to that math. But about 10 amino acids is the number of residues in the average alpha helix. Okay, I'm going to go ahead and end this slideshow so that I can open the next one. And it is 3:52. Okay, and I am recording. I always have a scary moment whenever I'm like, oh my gosh, I'm not recording, but I am. Okay. So this slide deck that we're looking at that I've got up is about 30 slides long. As soon as I open the first page, I'll then pull up your opener questions and polls. There we go. We got our openers. We got three of them. I'll give you a second to look at those on your own. Okay. So protein sequencing and analyzing or analysis of proteins. Now, common questions about peptides and proteins. Well, the first question is why? Why would anyone want to learn anything about a protein? Why would anyone be interested in a peptide? And I think that there's probably, I could, if I gave you guys a discussion and said, hey, why do you want to learn about a protein? Medicine. Yeah, it's a good reason. I think people would probably all kick up different responses. They'd be great answers, totally valid answers. Um, One that I want to share with y'all is introducing a technique. Um, could anyone add into the chat the two letters? It's an acronym for what is the point where a amino acid has an overall zero charge. I'm looking for two letters. There's an overall net zero charge, pi. Yep. The isoelectric point. Okay. Now, the isoelectric point is a point where your molecule is least soluble. The pi Well, an amino acid can have a pi. A protein can have a pi. A peptide can have a pi. Could everyone give me a three in the chat if, oh, okay, that makes sense. I hadn't really thought about that, but if an amino acid can, absolutely a peptide or a protein can. Okay. Now, is it reason when we were doing those pi calculations of individual amino acids, we weren't ending up with the exact same number. There were some PIs that were like 9.6 and then others that were like 3.45. So amino acids have different PIs. Proteins are going to have different PIs. Could everyone give me a five in the chat if, okay, yeah, I got you. Okay, wonderful. Now, I bring this up because there is a technique that has been around since I think it's in the mid 1970s. It's called isoelectric focusing. And this is a technique that you can use to separate molecules based on their PI. And whenever I say molecules, I'm saying amino acids, proteins, well, yeah, peptides. You can separate these different molecules based on their isoelectric point. In order to do this, and in order to do this to a biological sample, you're going to have a protein sample. So just imagine that you took like a blood draw or something like that. Are you going to have lots of proteins in that sample? Absolutely. Are those proteins going to be different from one another? Yeah. There's going to be lots of proteins and lots of different proteins. So it's going to be kind of a very complex mixture. Now, whenever you're doing isoelectric focusing, I'm going to put a star by that because that's this first technique that we're going to do. You can set up this gel strip. And this gel strip has these different kind of, you could think about it almost like plates, where it has a pH gradient. So it's going to have a range of pHs. And so what that means is, let's say that we have a molecule. I'm going to go ahead and I'm going to try to label this. This is our pH of 9 region, 8, 7, 6, 5, yeah, 4, 3. Okay, sorry, my scaling on my labeling didn't come out perfectly. And what we're going to do is we're going to mix in our protein sample. We've got some green protein. And unfortunately, proteins don't have colors. So whenever you do actually have to separate them in a research lab, you don't have that really option here. But we've got these three proteins: the green protein, the red protein, the blue protein, much like Power Rangers. We're going to apply an electric current. And you know what? These different proteins are going to start out over here: green, red, and blue. They're going to start out on the left-hand side of this gel strip. And what's going to happen is these molecules are going to begin migrating. They're going to begin migrating to the right. They're going to be migrating, and what they are going to do is they're basically going to stop whenever they reach their PI. So this green one, my green protein, I put in the 8 range. So it stopped there because the green protein, and I'm completely randomly assigning this, the green protein has a PI of, let's say it's 8.3. So it stops right there in that 8 range. Could everyone give me a Y if you understand that and you're good with that? Okay. Now, let's say that my red protein, again, I'm just kind of randomly assigning these. Let's say the red protein has a PI of 7. And then last but not least, the blue one has a PI of 9. Okay, so my red protein is going to migrate. And lo and behold, it's going to stop in this 7 range. And then my blue protein, it's not going to really migrate far. It's going to stop there. So I started out with all 3 of these proteins in this protein sample. Then I'm applying them to this gel strip where I'm going to separate them, and they're going to crash out, or they're going to stop because they've reached their isoelectric point where they have a charge of 0, and basically they're stuck there. Could everyone give me a Q in the chat if, okay, I follow you, or a Z in the chat if I'm a little mixed up? Okay, I'm seeing lots of Qs. I did see somebody put an A, which is smack dab between the Q and the Z on the keyboard. Uh-oh. Okay, so I'm going to take that as I'm kind of there. Okay, so we loaded these proteins, and then we applied this electric current. The proteins began moving, but as soon as they hit their pi, they stop because they're insoluble there. Think about it as it's almost like they crystallize right there. They just stop. They can't move any further. That is known as isoelectric focusing. We're separating these proteins based on this principle, based on their isoelectric point. That's the point of this. Now, I'm going to end the poll because everyone's participated. I'll come back to those questions later. This technique that we're doing at the top is isoelectric focusing. We're going to separate them based on their overall charges. We don't know anything about them but their overall charge or their pi. Now, this technique that I'm illustrating here as a why would you want to study proteins and why would you want to study a peptide is because after you've done that, you can load them on this right here. I'm guessing that some of y'all have probably done DNA gel electrophoresis or another type of gel electrophoresis. Could anyone tell me when you, oh, I got a loud nope. Okay, so if you've never done gel electrophoresis, well, I understand. Don't worry about that. Gel electrophoresis is a technique on which you separate based on size. So gel electrophoresis, like DNA gel electrophoresis. You're going to separate based on the size of the piece of DNA. The larger pieces of DNA are going to get stuck at the top of the gel. So the larger stay up top and the smaller migrate to the bottom. So applying this to proteins, if you have one protein that's 100 amino acids and then one protein that's 1,000 amino acids, the protein that's 100 amino acids long is going to migrate a lot faster than the one that's 1,000 amino acids. So we got our 100 and then our 1,000 up here. So this technique that we're doing in total here is known as 2D electrophoresis. Two dimensions. You're separating molecules based on two dimensions. The first dimension is pi. The second dimension is molecular weight. Okay, Alexander, you added a nope on the haven't done electrophoresis. Would you mind? I think that's enough for what I need you to know. Just based electrophoresis is a technique where you separate based on molecular weight or size of the molecules. If you or anyone else have questions about electrophoresis, please don't hesitate to either email me or send me a private message in class. I I apologize if I was like, yeah, everyone should get this. I wasn't intending to sound so pompous, so I apologize for that. Um, so we're separating first based on our isoelectric point, so charge, and then on our second dimension, we're separating based on molecular weight. So it's a combination of isoelectric focusing and DNA electrophoresis, not DNA electrophoresis. I was using DNA electrophoresis as an example where you separate based on size. You can do what's known as protein electrophoresis where you separate based on size as well. So we'll come back to that in just a little while. But ultimately, this right here, this gel at the bottom. Now that gel at the bottom, I don't know if you ever watched the TV show Dexter. I remember watching it a long time ago. It kind of looks like that, where he's a blood splatter analysis. That's what that kind of looks like. But this right here, this gel at the bottom, is a separation of tons and tons of different proteins. On this x-axis, they're separated based on their PIs. And then on the y-axis, they're separated based on their molecular weight. So you could pinpoint something and highlight something right here and say, oh, okay, well, that molecule that I've got a circle around that has a PI of about let's call it 8, and a molecular weight of, let's say that this is 190 amino acids. Okay, then you could circle something down here and say, okay, well, that molecule has a pi of approximately, let's call it 4, and it is a protein made up of 75 amino acids. Okay, so there's okay, so that's kind of cool. You can separate based on these two different things, but more than that, this right here is a tool that was kind of originated in the 1970s, and then in the early 2000s kind of regained some steam because what some researchers were doing were they were taking someone who was a wild type for a particular type of cancer, and then someone who is had, I'll just say, cancer X, and they take two different cell types, and you do this combination of techniques, and you'd say, okay, what is the difference between electrophoresis and IR spectroscopy? Okay, so electrophoresis is a way of separating based on molecular weight. IR spectroscopy, and someone who's an organic chemist is going to be able to help me with this. That's a technique, infrared spectroscopy. That's where you're looking at functional groups. You can define functional groups on a molecule. If I'm wrong on that, I apologize to all the organic chemists in the house. Um, so, yes, 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 take that. Okay, so we've got the application of this technique. We've got wild type cells, and then we've got cancer X. And you do the exact same thing with these two different types of cells, and then you have different patterns. Okay, so here's all the proteins in the wild type cell. And then in the person who has whatever type of cancer, you look at theirs and you overlay these two gels and you see, okay, what's similar, what's different about them? And the point is that this is a way that you can look at two different cell types and say, okay, well, this is the person who is healthy, and this is the person who has this cancer, who has this disease state. When we overlay these gels, hey, wait a minute. This right here is absent in the wild type cell. What is this? Why is that there? Is this type of cancer causing some sort of protein breakdown? Is this type of cancer, what is this leading to that caused this protein to occur? Is this protein doing damage, or is this an artifact? Or is this just sort of collateral damage? Now, this is my very long-winded answer of answering the question, why would you want to study a protein? So that you could identify, well, possibly drug targets, possibly an explanation for why someone is in a disease state. Now, some more direct questions. So I was giving this as an example to describe techniques and how we analyze proteins, but also give a little bit of context for why we'd want to. So I hope that you guys remain familiar with isoelectric focusing, and we'll get more into protein electrophoresis in a little while. But 2D electrophoresis is using two dimensions or two properties on which we separate proteins. Okay, when it comes to what's important overall is to understand these techniques and the properties on which you're separating. Absolutely. Thank you for asking the straight up question. Um, okay, so some common questions that people want answers to whenever you're studying a protein. What's the sequence of the composition? So the composition would be like, is it 90% non-polar amino acids, 10% polar amino acids? The sequence is, well, what's the primary structure? Some more questions along those same lines is, what does the protein look like in three dimensions? can I visualize that protein? And does it look like a glob or does it have some sort of structure, organized and ordered structure to it? And then this native fold, these two things are kind of, to a degree, they're kind of similar, the same to one another. How does this protein achieve its biochemical role or function? And how is that function regulated? Then, how does it interact with anything else? And how is it related to other proteins? And where is it within a cell? These are all really important questions that we want to answer when we're studying a protein. So if you were to go to graduate school and you're like, I'm going to study proteins, I'm going to study this protein in C. elegans. Okay, great. These are all the questions that you want to be able to answer. What are the amino acids? What does that protein look like in 3D? What does it do? How is what it does regulated? Where is it? How does it interact with anything? These are kind of the who, what, where, when, and why about proteins and peptides. Now, polypeptides contain different amino acid sequences, and the sequence and arrangement of those amino acids gives a polypeptide its particular character and its properties. Is it a protein that is largely hydrophobic? Does it have a positive charge, negative charge? Is it overall neutral? What are some of those important details about it? Some polypeptides bind specific targets, which you can use to kind of fish them out of a very complex mixture. And that's one of the things that we want to do to understand how we can separate proteins. Now, we've already talked about one property that is important, and that is the PI. If you can adjust a solution to your protein's PI, to its isoelectric point, it's going to be insoluble. Everything else is going to be soluble. So if you got rid of all the soluble stuff and you're left with only the insoluble stuff, that's what you want. So that's one thing to think about when it comes to protein separation techniques. Now, this right here on the left-hand side, I want you to look at that as, this beaker with, you could think about it as marbles or candies or whatever. I always like to use M&Ms and Skittles and Reese's Pieces as kind of your different proteins. So let's say this beaker on the left-hand side, let's say that you wanted to get the red marbles. If you look on the left-hand side, let's say that there's 5 red marbles. And there's, let's say, just for the sake of argument, there's 175 total marbles. 5 out of 175. If you want to enrich for the red marbles or purify those red marbles, have you purified or enriched for those red marbles when you move from the beaker on the left to the beaker on the right? Have you, basically, you could think about it as if you blindly put your hand into this beaker to pull out a marble, you've got a 5 out of 175 chance of getting a red marble. Would everyone agree with that? Wire N to the chat. Okay. Now, if somebody made some changes or if somebody helped you out and they said, okay, now go to the beaker on the right, blindly put your hand in that beaker, what are the chances that you're going to pull out a red marble from the beaker on the right? Well, we're looking at about 5 out of, I don't know, just let's call that 20. Have our odds increased? Yeah. So another way of saying that is that we have enriched for our red marbles. We've basically increased the likelihood that we're going to get the marbles that we want. Now, how do you get from the beaker on the left to the beaker on the right? Well, if you had some sort of magic wand that you said, okay, now make all of the green marbles disappear. Okay, cool. Yeah, that would work. If you had some technique where you could say get rid of all the cerulean marbles. Great. Got rid of the cerulean marbles. Get rid of all the white ones. Gone. Yeah, that's great. That's essentially what you're doing when it comes to purifying a protein. The terms that we use to describe this, however, we use activity and specific activity because we're looking at your protein having a particular function and being able to monitor that particular function. Here, our function is we see a red marble. But in terms of a protein, it might be capable of doing something. It might be able to take reactant A to make a product, break some bond, make some bond, something like that. Okay. Now, I'm going to jump ahead a little bit. And I want you to look at this protein gel, and we're going to come back to it. We're going to come back to it after this slide right here. So, what we are trying to understand, and what I want you guys to leave today's lecture with, is I want you to understand some of the basic steps for purifying a protein. So, taking a very complex mixture, much like that beaker with all of those different marbles, and finding a way to select for the red marbles. So, a purification would be get rid of all of the non-red marbles and hold on to just the red marbles. Now, at each step in a purification, you want to understand and be able to quantitate your total protein and evaluate whether or not your technique was effective. Now, if you think back to like Gen Chem 1, Gen Chem 2, have you done separations before? Yeah. If you think Gen Chem 1, you do a decantation, you do a filtration. These are separation techniques. In organic, um, have you did you do any sort of column chromatography? Yeah. You separated based on polarity. Well, with biochemistry, we also do types of chromatography. We're not necessarily separating based on polarity, we're separating on other properties, though. Now, at each step, you want to evaluate whether or not the implement that you used was worthwhile. Was that separation technique good? You want to have an idea of what's the protein that you're interested in purifying. So if you can think about that red marble as the thing that you want us to fish out of there, or in terms of a protein, an enzyme, something that can catalyze a reaction. Okay, so what I'm going to show you here is a table for purification of an enzyme, a hypothetical enzyme, from a crude extract. Okay, so what I'd like you to do is first look at this very first step. You are going to start out, this is basically the bucket of marbles, or the beaker of marbles. It's the beaker of all of the marbles. Is your red marble in there? Yes, it is. It is one of the marbles that's in there. Now, that beaker has a total volume of 1.4 liters or 1,400 milliliters. Then you've got the total amount of protein in that beaker. So, how many marbles are there? Then, activity, which is those red marbles and essentially what they're capable of doing in terms of like an enzyme that catalyzes a reaction. Last but not least is your specific activity. So you have your crude cell extract, which is what you've taken out of you've ground up some E. coli cells, all of the proteins that are in all of those cells. Does everyone understand, or could you give me a one if you understand what the crude cell extract is? Okay. Going to ask lots of confirmation on this one because I have a tendency to explain something and then be like, where did I lose you? And so I don't want to lose you guys. Okay. So we've got our crude cell extract. That is all of our proteins ground up. We have one protein that we want to extract out of there. Now, we can detect that protein. And we have this total amount of, we have 10,000 milligrams of protein. That is all of the proteins, ours plus everything else in there. The first step that we're going to do after this crude cell extract is an example, or one thing that you can do is to precipitate proteins with ammonium sulfate. Now, that's going to kind of randomly precipitate proteins, and you're going to just kind of operate under the idea of it might precipitate my protein, it might not. This is where you basically add ammonium sulfate, and some proteins fall out of solution. After that, you're going to see the total volume of that mixture go from 1.4 liters to 280 milliliters. The total amount of protein went down. Oh man, is that good? Is that bad? Arguably, the total amount of protein, so specific. Anna, I hope everyone sees your question. So specific activity is just for our desired protein in even in the crude extract. Yes, it's a great question. So we're seeing the total protein from step one to step two. Yeah, the total protein went down 70%. Ooh, that seems like a lot. It is. But does that matter? Not really. Because what we are concerned with on this entire table is the specific activity. And as long as our specific activity is going up, that means whatever we have done has been a good or effective separation technique. And I'm going to take you back to the marble slide. Total protein is all of the proteins in that beaker. Activity is 5. First, we were at 5 out of 175, and that was our specific activity, 5 divided by 175. Then we did something and did a series of steps, and we got to 5 out of 20. Well, 5 out of 19, you'll see in just a moment. But our specific activity has gone up because the amount of our protein has not changed relative to the total protein. So what we're looking for and what we're wanting to see is specific activity go up because that's kind of our measure of purity. If your specific activity has gone up, you have enriched for your particular protein. So if there's one thing that I want you to take away from this slide, it is that statement right there. Specific activity goes up, that is a great thing. That's what you want to see. That means you are enriching for your protein. Now, it might be daunting to look at total protein. We went from 10,000 milligrams of total protein down to 3 milligrams. Oh my gosh, we lost so much protein. Yeah, we did. But in a cell, most of what you're finding is a bunch of junk. No disrespect, but if you're wanting to reduce your system down to one particular protein, and there's 25,000 different proteins and millions of copies of those different proteins, you got like nearly 25,000 proteins you don't care about. Does that make sense? Give me a 7 in the chat if that makes sense. Okay, Lou hit us with a 67. Okay, so after that ammonium sulfate precipitation, that's where we get into our different chromatography techniques. Ion exchange, size exclusion, and affinity chromatography. Now, I want to take a step back to show you, to kind of give you a visual here. I blocked out a pair of the columns on this gel because they're not really completely important or relevant to this. This lane right here, the soluble crude extract, that is all of the soluble proteins. The protein that you want to purify after doing an anion exchange and cation exchange chromatography column is right here. This one very robust band. Now, if you're looking at this and you're thinking, what the heck am I looking at? You are looking at a gel that has proteins that have been stained with a purple dye. So all of these bands, like these, those are all individual proteins. Then when you see something very robust like this, that's not to say that that is a particularly large protein, but instead that there is a lot of that protein. Now, if you look at this very last column right here, the purified protein, what I want you to recognize is that there's nothing up here in this black box that I drew, and there's basically nothing down here. So that means the king's ransom of the protein that you have is your protein of interest. So this column right here suggests a high specific activity because you have a very pure protein. Okay. Now I'm going to go back to this purification assessment because what I'd like to do now is go into these different techniques, specifically the chromatography techniques. Now, like I said, your purity or your specific activity is kind of a measure of the purity. The higher the specific activity, the more pure your protein is. Now, proteins can be separated based on a number of different properties. Like I said, in organic, you separate things based on polarity and lack of polarity. You use those chromatography columns that have a silica gel in them. Separation in a biological system, you can separate based on charge, size, or molecular weight. Affinity, so how much does something want to bind to something or bind to something else? Solubility, hydrophobicity, thermal stability. These are all different options or types of chromatography that can be used. Chromatography is commonly used for preparative separation, in which the protein is often able to remain fully folded. What that means is that whenever you do a particular type of chromatography, you can collect that protein off of the column, so you can continue using it. It's not like you have to destroy the protein to get it to stick to the column. Now, this kind of rundown of column chromatography is basically that you have a reservoir that has your protein or your protein sample. You're going to use a peristaltic pump to load that protein sample onto a column that looks like this. There is this matrix, this solid porous matrix that's a bunch of insoluble sugars, and those insoluble sugars have been modified in some form or fashion. And as your protein or proteins migrate through this column, they're going to begin to separate based on their, well, affinity for that column. Now, this is just sort of general chromatography. If the proteins have no affinity for that column, they're going to go straight through. If they have a great affinity for that column, they're going to get delayed a whole lot. Now, this is a nice visual because we've got this idea that, oh yeah, we can separate. I'm going to collect the green proteins. Oh, I need the blue proteins. I'm going to wait for those last. Proteins rarely have colors. They might if they're bound to like a chlorophyll or something like that, but other than that, they don't have color very much. So really, you're watching a clear solution move through a clear column. And it's a little bit demoralizing, frustrating, but that's that. Now, with that in mind, what this is showing, this 1, 2, 3, 4, and 5, that's showing a time lapse. So that column number one there, that is your protein, or that is whenever it's initially loaded on there. Five is after time, after, 10 minutes or something like that. The protein and the sample has run through there. Which protein is going to come off of that column first? Add it to the chat. C. The last one to come off the column is A. It's going to take more time and you're going to collect little fractions. These fractions down here, this would be 1, 2, 3, 4. Those different fractions are different volumes, approximately 1 milliliter, 2 milliliter, 3 milliliter, 4 milliliter, and so on. You can adjust those as you're doing this technique. Now, let's talk about our first type of column chromatography, specific column chromatography known as ion exchange chromatography. Ion exchange chromatography is a separation technique based on charge. There are two different types of ion exchange chromatography, cation and anion. Now, I hope that you guys can appreciate this comparison or analogy, but I always think of cation exchange chromatography and anion chromatography, and I'll just call them cation chromatography, anion chromatography. It's much like fishing. When you go fishing, you're trying to catch a fish. When you do cation chromatography, you are trying to catch a cation. Now, I am not a very good fisherman or fisher person, but typically when I go fishing, I use worms to catch a fish. I'm not yet into the point where I'm using fish for fishing. But what I'm getting at here is to catch an anion, you have to use a positively charged thing. So whenever I ask the question, what is the charge of an anion chromatography column? What's the charge of an anion chromatography column? Jada, you're absolutely right. It is positively charged because you're trying to catch an anion. What is the charge of a cation chromatography column? Negative. Absolutely. So, with that in mind, since this is sensitive to charges and proteins can have charges because of amino acids having charges, you're going to have to play around with, like, whenever a protein gets stuck to the column, you need it to elute. So the way that you're going to do that is by adjusting the pH of a solution that's running through that column. Does that make sense? Because if you remember when you do chromatography in organic, it's just like, well, wait, the more polar substance is going to take longer. The less polar substance is going to take longer. In here, if you have a negatively charged protein bound to a positively charged column, it's going to stay there almost indefinitely. If you adjust the pH of this solution here running through that column, ADJ, adjust pH, well, then the proteins will release at a different point or earlier. So you have to adjust the pH of the solution that's running through the column. Now, one of the things that I want you to think about is if you have a positively charged bead, you're going to be getting negatively charged proteins that adhere to that column. And whenever I say bead, I'm referencing this gray thing is this gray bead right here. So a negatively charged gray bead is going to attract positively charged proteins. Now, that's one of the reasons that I spend a lot of time on amino acid R group structures and classification of polar positive, polar negative residues. I want you to know that. So if you have lots of, or thinking ahead to if you have a protein with lots of aspartic acids on it, on the surface, lots of aspartic acids, will that adhere to a cation column or an anion column? Aspartic acids, aspartic acid R group looks like this. So that will adhere to a resin bead that is positive. Can everyone give me a W in the chat if that makes sense? Great. Okay. Happy to read that. Now I've got a poll question that I'm about to share with y'all. It's on the next slide right here. The question reads, which protein would most likely bind to a cation exchange column? Remember, a cation exchange column is a cation column. So what's it looking for? And remember, we're thinking about the R groups alone. Oh, popular answer is correct. Oh, I spoke too soon. No, it's still good. I'm gonna give another 10 seconds or so. Actually, I'm just gonna start talking about this one. A cation column, well, you're fishing for cations. Which of these are going to have a positive charge? Leucine? Nope. Alanine? No. Those are two non-polar residues. If you know the classification of your amino acids, you can eliminate those right off the bat. So then you're left with a protein rich in histidines or a protein rich in aspartic acids. Now, those aspartic acids, those are more than likely going to have a negative charge because they're polar negative residues. Histidines are going to be polar positive. So the correct answer is B. For B is the correct answer. So histidines. Okay. Now, if I were you, I would take this. Petra, do you have a question? I did see this.
Yeah, yeah, wait, I'm confused. I thought so if a protein would most likely bind to a cation exchange column, so does that mean that the protein would have to be negative? Because I thought you had to use to catch a cation or
Okay.
I don't
This is okay. I just got two private messages where someone's basically expressing the same thing. Okay. A
okay.
cation column is going to be very effective for catching cations.
Okay.
So then I think that let's one way that you could think about it is this cation column is packed with, I'm going to switch back, with lots of beads right here, these gray beads. So the question is, what's the charge of those beads? In a cation column, I'll write this cation column has negative beads. So with a cation column having negatively charged beads, you're going to be attracting positively charged proteins or positively charged amino acids within a protein. Does that make sense?
Yes.
Okay.
Yeah.
So
Thank you.
anion would have positively charged. Ernest, absolutely. So in the event that you're like, no, that doesn't make sense, um, it's kind of tough to like wrap your mind around it. That's why I hope that it's helpful, but I always think about When I'm fishing, what am I looking for? I'm trying to catch. I'm naming the thing that I want to catch. So when I'm doing cation chromatography, I'm trying to catch a cation, so a positively charged thing. How am I going to do that? Well, I have to use the oppositely charged thing. And so that's giving me the description of my beads or the charge of my beads. So, so cation exchange chromatography, anion exchange chromatography, your proteins are going to adhere to those beads. And in order to get them to release, you adjust the pH and boom, they're going to come on. Is it like a cation is negative, then you catch a positive, and an anion is negative, so you catch a positive? No, a cation, you have to think about, or what I think is helpful is to think about the beads themselves or the protein, because we're trying to capture a protein with a positive or a negative charge. So I like to say cation column. Now I'm trying to come up with like a description of the column. This column has beads within them. These beads are insoluble sugars. These beads are negatively charged. They bind positive proteins. An opposite of that is an anion column. What are my beads? My beads in an anion column are positive, and they will bind negative proteins. Cation column catch send a private message. Cation column equals catch cations. An anion catches anions. Yes. Okay. Now, okay, great. Next up is we've got two other types of chromatography. Then I'm going to put the closer questions up. These two types of chromatography, absolutely. Thank you very much for asking clarifying questions. Whenever, yeah, we spend some more time, if that helps 5 more people, that's worth it. If that helps 1 more person, that's worth it. Okay, so you can also separate based on molecular weight or size. This slide or this one, Natalia? This slide right here, I presume? Okay, perfect. Great. Okay, so size exclusion chromatography. This is also known as GF or gel filtration. This is a technique that's a little bit confusing because of the term gel, because people then think, oh, gel filtration. This is the same as gel electrophoresis. It is not. Don't fall into that trap. Gel filtration chromatography is a separation technique where you can separate based on the size of proteins or the size of molecules. Now, if we look at this column up here and down here, what I want you to notice and think about is which of these proteins are going to come out of the column first, the larger or the smaller ones, L for larger or S for smaller.
I remember doing this.
Well, the ones that are coming out of the column are down here. We've got these green balls representing the larger proteins. So in gel filtration chromatography, the larger proteins elute from that column first. And that's always very confusing. If I can, if you don't mind, go with me on this adventure. Let's say that you had a, let's say that you had a solo cup, a red solo cup, and you had the bottom of it cut out. So there's no bottom to it. If I tried to put a basketball through that cup, is it going to be able to go in there? It's not. So basically, what I would do is I'd take that basketball and I would kind of try to slam dunk it. Oh, it's not going to fit. So I'd roll off the side. Okay. So that doesn't fit. That's the large protein. A small protein. Let's say that we got a marble. First of all, do you think that the marble could fit through that solo cup? Yeah. Now, that marble, what it's going to do is it's going to touch the side and it's going to be in contact with the side of that solo cup. So it's going to go all the way through it. So it's going to be in contact and it's just rolling around. It's almost like um, I don't know, like one of those things that they have at children's museums, science museums, where you drop a penny and it goes concentric circles. That's what this marble is going to do. It's going to be in contact with the entirety of that cup the whole time. Now, if we compare that to, so we just did a marble, let's say a golf ball. The golf ball is going to fit through and it's going to do the same thing. Which one of them, the golf ball or the marble, which one do you think is going to make more rotations on the interior of that cup? The marble, because it's going to go, it's going to take so much more. And it's going to, maybe do like 30 spins around it. That is the small protein. The small protein is going to be in contact with these beads. Now, the beads here are an insoluble sugar, but the difference is look at this right here. They have these little openings in them. And in those openings, in those grooves, your proteins are going to enter those grooves and they're going to be in contact with the side of them, much like that marble was in contact with the solo cup the whole time. So we had the basketball. That was the big protein. It didn't even fit in there. Then we said, okay, well, that doesn't fit. It's going through, it's going to the bottom first. Then we had a golf ball and a marble. Which one of those is going to get through first? Well, the golf ball, we put that in the solo cup. It goes 1, 2, 3, it's done. It's through that solo cup. The marble goes 1, 2, 3, 4, 5, and so on. And then it's through. So the marble is smaller and it makes its way through slowest because it makes so much contact with the interior of that bead or the interior of that solo cup. Now, I'm going to yeah, okay. I'm going to put the closer questions up. Unfortunately, they talk about something known as SDS page. That's our protein separation technique. And there are some things about SDS page that I didn't get to today. On Monday, we're going to pick up with separation based on affinity. Well, we'll take off with separation based on size, and then we'll do affinity. Um, Go ahead and answer the closer questions best you can. And then we'll pick up with some of this stuff on Monday. And then we've got our test a week from today. I'll be sending out some more information about the test. But in the meantime, after you answer the closer questions, I hope you have a great day. I hope you have a great weekend, and I will see you guys on Monday. Thank you all very much for coming to class, and thanks for your questions and your participation. It means a lot to me.